right, now we're going to move on to dihybrid crosses. So going to go back to artificial populations, things that Mendel looked at. And he also looked at what were called dihybrid crosses. And so this requires the analysis of two characters, and each character has to have at least two pairs of contrasting traits. So again, what do we mean by this? So we did the monohybrid cross, and we said that the parents differed only at one character. And so now we have parents that differ at two characters. So the seed shape. So in one case you have a smooth, and in the other case you have a wrinkled, and then the seed color. So one is green and one is yellow. So there's two different characters that are distinct between these two parents. And so this is a dihybrid cross. So again, they're true breeding, and true breeding just means that they are homozygous. And based upon our knowledge of which one is dominant and which one is recessive, we simply have indicated that on the genotype information here. So the green P, that's the recessive color, the recessive trait, and then the round is the dominant. So this individual can only produce one type of gamete. So it has to produce a big R and a little y. So it only has one version of the R gene and one version of the Y gene. This individual can also only produce one type of gamete, so the little r and the big Y. And so because of that, all of the F1 individuals, again, are identical to each other. And all of them have to be big R, big Y, little y. So they're all heterozygotes. And so all of them have the same phenotype, but one thing you'll notice is now they don't share the phenotype with one of the parents. So this individual is round and yellow, which does not match either one of the parents. And that, in part depends upon exactly how the initial combination was. So these individuals can then interbreed with each other. And so in this case, if they interbreed with each other, you have to worry again about how many gametes that each individual can produce. So these individuals can produce 4 different types of gametes. So big R, big Y, big R, little r, big Y, and then little r, little y. And each one of these has a 1/4 possibility of being produced. Each equally likely. That's one of the assumptions of Mendel. And so as a result, the intercross of these F1s produced this new set of ratios of phenotypes. It was a 9:3 to 3:1. So 9/16, so you'll notice that it's a 16th, it's out of 16th. So 9 out of 16 have round and yellow. 3 out of 16 are round and green. 3 out of 16 are wrinkled and yellow, and then 1 out of 16 are wrinkled and green. So the most common type of phenotype is the one that has both dominant phenotype, so the round and the yellow. The least likely had the recessive of both. And so how did he do this? So how do you figure this out? So this comes from the ability to produce 4 different gametes. So this is one of the things about genetic analysis that you need to look at. Is one of the things you need to look at is the ratios. So before we saw it was 3:1. So in the monohybrid cross, it was 3:1. And so these were all ratios of 4, 3 out of 4, 1 out of 4. And now for the dihybrid cross, it comes out of 16. And so this can give you a clue, for example, of how many different gametes the parents produce. So here we see that each parent can produce 4 different gametes. Each person can produce 4 different gametes. So parent 1 is on the left, parent 1 is on the right. And so 4 times 4 is equal to 16. And so this is part of our clue of why this ratio is out of 16, is because each parent's produce 4 different genotypes. So we can do a Punnett square. And so the Punnett square simply allows us to show the 4 different gametes that can be produced. So here 1, 2, 3, 4. Here 1, 2, 3, 4. Those are all distinct. And now what happens if this sperm fertilizes this egg? So in this case, we just simply combine them together. So the genotype of the offspring would be big G, big W, big W. And then we can just fill in the phenotype based upon that. This individual right here, little g, big W. And if it combined with little g, big W, then this individual would be produced. And so we can group these all together. So for example, these 2 squares have the same genotype. These 4 squares have the same genotype. This one, and then this one and this one have the same genotype. So you can also look at this from the genotype way. And so there are 1, 2, 3, 4, 5, 6, 7, 8, 9 different genotypes that can be produced in the F2. So it'd be a 1 to 2 to 2 to 4 to 2 to 1 to 1 to 1 ratio. And if you group these together by the phenotype, then you would say these are all the 4 genotypes that produce the same phenotype. So all 4 of these genotypes would produce yellow round seeds. These 2 genotypes would both produce yellow wrinkled. These 2 produced green round, and then this is the only genotype that have the green wrinkled. So for the dihybrid cross, it's a very important ratio to remember, the 9 to 3 to 3 to 1 ratio, and that's with respect to the phenotypes, and that can be figured out just by looking at this Punnett square. And you can even move on to more complicated crosses, so trihybrid crosses, and you would simply now, instead of having an individual produce 4 gametes, you would have an individual produce 8 gametes. So a trihybrid is when they differ at 3 different traits. So it'd be something. So the F1 individual would have to be heterozygote for 3 different genes. And so this individual would produce 8, and so you'd have a Punnett square with 64 boxes. And so the results in the ratio would be a 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1. So this individual would be the dominant for all 3 traits. So if we just listed it, dominant, 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 this one would be dominant, dominant, recessive. This one would be recessive, dominant, dominant, so and so forth. So these have all 3 dominants. These 3 have 2 dominants. These have 1 dominant, and these would be all recessive. And so again, the trihybrid cross, you would have this individual for cross to this individual. So there would be 3 different traits, 3 different characters where they're different. The A, whatever's controlled by A, B, and C. This parent can only produce 1 gamete. This parent can only produce 1 gamete. You have a heterozygote F1, and this individual could produce 8 different gametes. So why are these gametes uniquely able to be found? And that is simply because genes are part of chromosomes. And so when we look at the results of Chapter 2, so how are gametes formed? So the idea is that there are genes, and these genes have physical location on chromosomes, and different genes have different positions. So here we have a gene G, and it is on chromosome 1, and here we have a gene W on chromosome 2. So during meiosis, the tetrad forms. And so this is a heterozygote individual. And so you'll notice that this chromosome has 2 copies of big G. And that's because this was produced from DNA replication of this. So you have exact copies. And then same thing here. So there's little g, little g. And so during meiosis 1, there are 2 possibilities. So when we segregate the homologous chromosomes to different cells, this could happen, where the big G gets segregated to the same cell as the big W, or the opposite effect can happen, where the blue chromosome 1 is segregated with the orange chromosome 2. And so as a result, these 2 meioses would form different types of gametes. And so because these sort independently, it makes each possibility equal likelihood. And so that's why it would just simply be an equal probability.